37 million people have kidney disease. It may not be the same disease for all 37 million people. And so if we can apply some of these principles in terms of genetics and some of the things that were emerging, like functional genomics, maybe we could have better insight onto how to have better therapy.
Welcome to the show, Power to the Patients. I'm really excited to be interviewing Jason Coloma today, who is the CEO of Maze Therapeutics. Before Maze, he was a venture partner at Third Rock Ventures, where he was incubating and looking at early stage biotech opportunities before getting the conviction to come build Maze. So I'm excited to learn about that journey and what brought you here. But maybe I'll give you a second to introduce yourself.
Yeah, no, thanks, Brent. Thanks for having me. Yeah, no, I'm a trained scientist, went to grad school, kind of worked in drug discovery for many years, ended up Genentech before the acquisition. We ended up getting acquired by Roche. That was transformational on a few levels, including myself personally, because I was asked to move to Basel, Switzerland as part of that. So we were acquired in '09, went out there in '11, stayed there for a little over 5 years. And then, through that process, I ended up, transitioning, of course, more into the business. My last role was, the head of business development, primarily focused on oncology and research technology platforms. And that's where I got to know some of the venture capitalists through partnerships and working on portfolio companies, partnerships with them. I got to know the Third Rock people a little bit. And then when I moved back to the United States, I was fortunate enough for them to ask me to join as a venture partner, worked on a few things as an entrepreneur, and then really had the opportunity to build out Maze. And one day they said, "Hey, have you ever thought about being a CEO?" I said, "Not really, but why don't we give it a shot?" And here we are now, almost 7 years later, where I've been able to build it from scratch and have the good fortune to keep progressing the company to the point where we are today.
Look, Jason, when I speak to founder CEOs, I always love to understand what gave you the conviction to take a bet like this. It's a decades long commitment.
Yeah, no, I left pharma about 10 years ago. And I think what was really nice, Brandon, because I was primarily in pharma, mostly working in drug discovery earlier in my career. So Third Rock at the time, was really a great platform for me in the sense that, they had built some great companies, Myocardia, which eventually was bought by BMS, Revolution Medicine, which made some news over the last couple of weeks, Blueprint Medicine, which was acquired last year after being commercial stage. So for me, it was a little bit, just to draw the analogy of, sort of my academic training, it was like a postdoc in entrepreneurship, to be honest. The company had a good history and great people to be able to, how to be thoughtful about company build formation from the very beginning when it's an idea. I got to observe it. I got to help build a couple. And then I, really being able to partner up with some of the people here in the Bay Area office to really kind of build out what eventually became Maze. And so having that, a little bit of that, I wouldn't call it safety guard, but I would just say sort of having the resources of being able to walk around or at least get on the phone and of people who have built some really successful companies was tremendous for me coming straight out of big pharma. And that gave me sort of a confidence of incorporating the best I could, some of the lessons learned, some of the gotchas. You're not going to get all of that right until you actually get into the company yourself, as building a company. But it gave me a much better foundation, I think, than just jumping straight out of pharma, to be honest, for me. And I think that was tremendous. Had the opportunity to do that with some great people. One of our co-founders, Charles Homsey, who was also a co-founder at Myocardia. He was also a co-founder with Neil Kumar at BridgeBio. So obviously he was a trained clinician scientist. And to have people like that around that can really think about how do you apply some of the things we were trying to do from a genetic standpoint, but also keep in mind, how to think about this for patients. And the ultimate goal is to make a medicine, and to have drug hunters like that around us at the beginning was tremendous.
Yeah, absolutely. And let's get into the science a little bit. What did you see that led you to want to say, okay, this is a platform I want to build around?
As I kind of described at Genentech and Roche, I'd worked in, at the time, precision medicine, which is trying to identify the right patients with the right therapies and match that very nicely, primarily in oncology, right? We had a lot of work in oncogenic drivers. We did things like partnerships with eventual acquisitions like Foundation Medicine to really identify that. I mentioned Blueprint Medicines, which was also a targeted approach. And so, I kind of was inspired by, well, could you apply this principle, at least precision medicines, to diseases that hadn't seen it before, like kidney disease, right? 37 million people have kidney disease. It may not be the same disease for all 37 million people, right? And so if we can apply some of these principles in terms of genetics and some of the things that were emerging, functional genomics, maybe we could have better insight onto how to have better therapy, because there hasn't been a lot of innovation and clearly no precision approaches when it came to kidney disease. So that at a high level, that's what sort of inspired me to kind of work on this kind of concept. And Charles and a few people had come out of myocardia, Eric Green in the beginning, and some others that really were sort of, okay, we learned a lot, or at least in the context of cardiovascular disease. Could we apply some of these principles to kidney? We weren't sure. And diseases like kidney, could we really do that? And so we really started that journey in the process to say, okay, can we sub-segment 37 million people that have kidney disease into smaller segments where we might be able to use the insights in genetics and functional genomics to really guide, in this case, small molecule drug discovery, such that we can build better medicines for those patients, right? And so, a little bit of the learnings of how do you think about precision medicine? How do you go after something that could be, really transformative given the health impact that, if you develop a kidney disease medicine, it could be quite impactful, because beyond 37 million people having it, Brandon, it really is, a silent killer. People, it is now the ninth leading cause of death globally.
Yeah.
People don't really recognize that. Physicians and nephrologists in particular try to do everything they can to keep people out of dialysis, which I'm sure, you or others that are listening know someone that's gone through that journey. It's life-changing because of the impact it has on your daily life. And more importantly, your outcomes are horrible once you enter into dialysis. Like, we used to show this graph showing a Kaplan-Meier curve, a survival curve of someone who was in dialysis, 10-year survival curve versus one that had metastatic breast cancer. It looks very similar. And people don't think about that, right? Like, once you're in dialysis, your outcomes are horrible. And so a nephrologist will do everything they can. They said, my job is to keep them out of dialysis the best I can, right? Because when I do, once they make that transition, and if we can't get them back out of dialysis, we know that's bad, right? So if we can work on medicines, that allow us to really change the trajectory, maybe even be truly disease modifying, that has a huge impact given the, sort of the global health epidemic that's been created due to kidney disease. So that was the problem statement we started a long time ago. And, of course, what started to converge? Like the concept of, okay, everyone was saying, okay, they kind of settle on what's like a statistical association of a gene to a disease, right? And then they kind of go, oh, and as you might know, Brandon, like you could read about these all the time. Like they even make the New York Times, or, my mom will ask me like, oh, this one's connected to Alzheimer's. But the probability is that those actually transition from a genetic insight to a medicine concept is actually pretty low, right? And so what we wanted to do is move beyond the statistical association of a gene to better understand genes themselves in the context of kidney disease or disease like kidney disease, but more important to understand the variant, or each gene has many variations, which is just a different sequence. If we can understand the variants of genes in the context of a disease, then we can better understand function and mechanism of how that gene is causing the disease. And if we could do that, that will be better information that can guide a drug discovery process. That's fundamentally what we were thinking about. The initial papers that talked about variant functionalization, as it was called, were written out of the Broad Institute like 15 years ago. Like this concept of, can you understand genetic variation and functionalize variants of genes in the context of disease? A lot of great papers were written. The concept was there, but no one had really kind of implemented in a small company format. And two, you really didn't have the tools or the data to do it. So like, what changed over that time frame? Like, you had all these data sets now, like UK Biobank and others, that had genetic with paired clinical information in a longitudinal fashion of hundreds of thousands, if not millions of individuals. You can track that over time and look for that in terms of genetic variation. You had things like CRISPR, which, obviously resulted in a Nobel Prize for individuals. You had techniques like that, single cell genomics, better ways to perturb and understand the context of variants in this particular setting that didn't really exist, right? And then you had other things in terms of tools from a drug discovery standpoint that started to emerge, like cryo-EM. Can we better understand the structures of the genes and the proteins eventually of what we're trying to, sort of drug, AlphaFold, which also, helped win a Nobel Prize as well. Like these things started to emerge of like, there's new tools that allow you to, if you properly integrate them, you can move better from a genetic association ultimately to a drug candidate. Like that stuff didn't exist. And we were one of the few people at the beginning that were integrating these large scale data sets. We were building out variant functionalizational tools, including experimental as well as computational. And then we started applying some of these newer techniques, like at the time, cryo-EM, machine learning informed DELs and other things that allowed us to really be able to use that information in a more, I would say, drug discovery pragmatic way to build a molecule. And so if you look on our website and look through our S1, if people still have access to that, every program went through that process, right? We went through it, kind of described it, and all the things that we work on that are now in the clinic, as well as things that are preclinical or things that we've partnered, utilize that same technique. Go beyond a, statistical association, understand the variant of a gene in the context of a disease, functionalize it so you can better understand mechanism, build the molecule with the hypothesis in mind. And then, of course, what's nice is because we start with the genetics, we can try to identify the patients we think will have the highest probability of seeing an effect.
I'm fascinated to learn a little bit more about how the application of these techniques changes how you maybe like subsegment the problem, right? If the problem statement is kidney disease, which is quite vast, help me paint a picture of the framework now of the way you see the different types of kidney disease, the ways you maybe even further subsegment them, and where the applicability starts to come in.
Yeah, absolutely. So, let's take our lead program, APOL1 mediated kidney disease, as an example to kind of walk you through that overall, sort of framework. So APOL1 mediated kidney disease, for people listening that don't know, it predominantly affects the Black community. There's two particular genes that cause what we call a toxic gain of function, or a function that you don't want, that ultimately results in kidney disease. Okay, so people kind of knew the statistical association for years, over 10, maybe 15 years. The original genetics papers were written saying, oh, these variants seem to be correlated with increased disease and kidney disease. Well, that's interesting, but no one had figured out the function. Like, how is it actually causing kidney disease? And then you look through the literature, there's probably, a dozen, half a dozen to a dozen different hypotheses, but no one had proven it. We used our techniques to do a couple of things. One, we knew it was sort of these variants that caused the disease. They were just called G1 or G2. But we used our data sets to identify and in collaboration to find a protective variant. We ended up publishing, it was called N264K. And that's important because it basically, interesting enough, even if you had the variants that should be causing disease, if you had this protective one also co-expressed or expressed at the same time, the people weren't getting sick or as sick. Now that's interesting. Like, they were protected from a standpoint of eventually forming kidney disease. We found that, protective variant. It hadn't been, published before. What we did is we took that variant and brought it into the laboratory to try to understand, well, okay, if we can understand how APOL1, how it's protecting, maybe we can understand how APOL1 was causing disease, right? What's the function? And then we can use that information to think about drugging. So it turned out of all the hypotheses that were in the literature, what ended up when we functionalized the variant was that APOL1 was acting basically as this pore, but under certain conditions, they overexpress in your podocyte or your kidney cells. They basically overexpress, insert themselves into the membrane of the cell. What that means is they're basically punching holes in your kidney cells, right? And then unfortunately, there's no gate on it. So they allow certain things, in this case, these particular bad actors into the cell that eventually cause the cell to be damaged and even die. Wow, that's interesting now. Because now we know what actually is causing the nephrotox or the kidney injury. And so let's try to make a drug that basically prevents that. That seems simple, but like no one had known that. We actually figured that out. And so what you want to be able to do at minimum, Brandon, is, block the pore. You don't want the bad actors to get into the cell. And if you could, maybe you could prevent the pores from forming to begin with so that they're not punching holes into the kidney cells, right? And it turns out that's what we ended up building as a molecule driven by that whole process. right? And because we now have a molecule that is able to do that, now we can potentially see that benefit in patients. And then because we know which genes kind of cause the disease, we can then identify the patients that should best benefit from that. So it's the full arc. You go from an association to, wow, we think these patients would probably be best benefit from an approach now that we really understand what that variant is actually doing to cause disease.
How was this condition managed before we had this understanding?
So a little bit about the patients, right? Because that's important for viewers to understand. So, okay, so kidney disease is typically associated with an elderly population, right? They kind of think, they enter like very later in their age. Well, it turns out those that have APOL1 kidney disease, Brandon, they tend to be younger. So they clinically present or they find a disease before the age of 50 as opposed to much later in life. They end up progressing or they're sicker in the end. So what that means is they transition to end-stage renal disease or into dialysis, as I point out, pretty bad, about 10 years earlier than those that don't have the variant and have kidney disease. And then the other thing that we know now is that they're not responding to the current, options, including immunosuppressants or ACE/ARBs or other therapies that are considered the standard of care. They're not responding. So they're younger, they have more progressive disease, and they don't have a lot of great treatment options to date, right? So what we've learned, like in terms of the journey of the patient is, okay, so typically a younger person will go get their annual checkup, right? They realize they have high blood pressure for some unknown reason. Oh, that's interesting. You're younger and you have high blood pressure. Let's do a whole lab workup. So people will typically look at, your kidneys and your liver to see if something's wrong. You get that lab test back, and what they realize is that they have too much protein in their urine. That's a measurement. If you looked on your LabCorp Quest kind of report, it's UACR, and they'll see that it's elevated relative to the normal range. So a person that has, normal kidney function should have a UACR or protein in their urine of less than 30, right? Some of these patients could have like 2,000, right? Like really high nephrotic disease. So if you have so you have high blood pressure, you have some kidney function. So the primary care physician will typically, if you have a UACR or protein level in the urine that's greater than 30, they'll refer you to a nephrologist. Nephrologists will try to figure out what's going on. They'll usually just monitor for a while to see if they can get the protein. They'll give you an immunosuppressant, see if you can kind of monitor it, like lower it. If it's not effective and they keep seeing, increases in the protein, or if they see another measurement, which is called EGFR, which is basically a measurement of how well your kidneys filter all the toxins and bad stuff that people have. If they see that, you want your UACR to go down and you want your EGFR to go up. That's sort of like clinically what physician will want to see. And if they see the opposite, then they have to start putting them on therapies to try to change that, right? So now, of course, as I mentioned, the patients are younger, they're not responding. They start putting them on things like an immunosuppressant, don't see a response. They put them on maybe an immunosuppressant and an ACE/ARB, not seeing anything. So they keep having progressive disease to the point that, unfortunately, some of them transition pretty fast, as I pointed out, eventually getting into dialysis. So if we can interject, like all these therapies don't get to the underlying cause of disease, which us and other people working on APOL1 mediated kidney disease, okay, those things don't work. Let's go after truly what might be causing disease, APOL1 itself. And if we can do that, perhaps we can change the trajectory of kidney health for those patients.
My understanding of this condition, and check me on this, is that something like 15% of folks with the gene end up developing this type of mediated kidney disease, APOL1 kidney disease. Is the proposition that this is interventional before the development of the disease, or it's an intervention that you can apply after it starts showing?
I mean, so you're kind of highlighting for people listening. So yeah, around 13, 15% of the people. So say there's 6 million individuals that have have the gene. About 1 million of them will have the gene in some form of kidney disease, right? And then, of course, what we're saying is that probably, to now your specific question, you probably want to initially show that this can work in people that already have progressive disease. So it's not to prevent, it's actually, okay, can we try to improve the EGFR or the kidney filtration rate to go up and reduce the protein in the urine to go down? So that way we can elongate the time frame of their progression and eventually and try to hopefully prevent them from going into dialysis. I think over time, one could imagine doing something more preventative, right? But that, from a development standpoint, would take longer, be a larger study. So I think
Yeah,
what you initially want to do is try to help the patients that have, more aggressive or more progressive disease.
makes total sense.
Yeah.
One of the topics on the show often is the challenges in R&D and conducting research, as you're starting to hint at here. I imagine that this kind of subset is not well diagnosed or not universally diagnosed. Like, tell me how you think about designing programs that even support patients in this population. How would they even know that they might be a candidate for this?
Yeah, absolutely. I think, as you're pointing out, I think that's one of the challenges where, trying to apply genetics to a disease like kidney disease, right? I think that is not clinical practice today, right? You don't go into your primary care and then say, get a battery of genetic tests and go, oh, I have this. Should I do something about it? That's not how they're treated. They had this more elongated roundabout way to figure out, oh, wow, you have the gene. We should do something about that. Right. So I think, part of that is, and I think, multifold in the sense of awareness. What's interesting when we started this, because some of us had worked in, probably disease areas where patient advocacy is more prevalent, rare diseases, oncology. You know, one of the things we were, I wouldn't say worried about, but probably kind of exploring is what's the role of patient advocacy, public-private partnerships, that didn't seem very mature in kidney disease when we started this journey. What's nice to see now, Brandon, is that's actually increased quite a bit. There are groups like Nefficure that have kind of done this, kind of making APOL1-mediated kidney disease more, awareness programs and suggesting people should at least try to, if they have kidney disease, get genotyped. There are more public-private partnerships like the Kidney Health Initiative, which we're a part of, that allow for more education. And then there's academic working groups like Parasol, which has been interacting with the ecosystem, including the FDA, to talk about what are the right surrogate endpoints to think about more accelerated approval pathways for people with diseases like APOL1 kidney disease. Right. This stuff didn't exist 10 years ago. And I think that's what's really encouraging is that, you have more patient advocacy, you have more awareness, you have more public-private partnership, which is putting a big lens on this, and academic working groups that we're also a part of that really sort of help with this. Okay, so that's one level. Next is like testing infrastructure, right? So so what's good is there's been progress there too. So this is now a standard genotyping test, mostly blood. It could be urine too, but it's on the standard LabCorp request panels. So a physician, a nephrologist can actually request that. Now that didn't exist before. So that's good. And then I think the third thing, which is important, is just being able to sort of engage physicians, classification of the disease. So what that is typically an ICD-10 code for those that aren't familiar. It's just how you classify a particular disease. For a physician, it's very practical because now they can actually bill the testing, and get reimbursed for the testing eventually, which is important. So I think we've seen inroads in all of that, right? It's still early. A lot of work has to be done in order to, continue that. But what's promising is even early in development, you're seeing this as opposed to other diseases where we've worked on, where it takes even longer to get to that level of infrastructure, where you have advocacy groups, you have testing on standard panels, more awareness, and then you have an ICD-10 code, which actually helps and trying to drive, sort of testing at the physician level. And so I think that's all great. But a lot of work that needs to be done. But I think with more therapies available for those patients, I think you can imagine that increases over time.
Yeah, certainly. We've seen the progression of biomarker testing in oncology becoming more prevalent and finding its way into standard of care, and just changing the way that patients and physicians engage on all that.
I think the other angle of this is, beyond patient advocacy, it's how developers like ourselves, like how do you engage the community and partner with the community to make this aware, to drive sort of awareness, right? I think we're finding that something that's a little bit different I was used to coming from big pharma is how much I would say med affairs work, patient advocacy group work that happens even in early development in order to, sort of drive awareness as well as to help drive clinical development. That's unusual, I think, in a lot of different therapeutic areas. I think when it comes to APOL1 kidney disease, this is important. We've had people, sort of our engagement. We actually have patient advocacy, people here engaging with the broader community, not just nephrologists, but even other parts of the community. We have a patient advocacy advisory board. One of the members is actually going to be here next week to meet with us and the staff. We've had people from the community, including one of the pastors of one of the largest congregations in Philadelphia, come and come meet with us and talk about, sort of what would be the appropriate thing to do since we're mid-stage development in terms of engagement models with the community, which, as you can imagine, in oncology development, we didn't need to do that, right? So this is a different game, but I think
well, simply said you're trailblazing with this.
Yeah.
Yeah.
Yeah, us and a couple other sponsors, I think, are, really being able to help drive this and be thoughtful about how do you have the right level of engagement? Because you don't want to overpromise. You're, working with patient advocacy groups all the time. You can't overpromise because you're in mid-stage development. So how do you authentically do that with people such that, they also see that, okay, and how do you build trust? Because it does take trust building over time in order for them. They're not just going to say, oh, yeah, please go talk to all my congregation in my church. Like, that's not going to happen. Right. But over time, we can start to establish those relationships and that level of trust such that, we can have an authentic conversation about, okay, why this is important at the end of the day, even just to get genotyped.
And to your point, especially with late or early stage, mid stage development, I find that part of building trust is outlining the fact that some of this is inconclusive and it's still, we're still asking questions. And in a lot of the patient conversations we have with our community, the kind of core message that resonates is, hey, like, help us ask this question together.
Absolutely. I think that's an important point. There are no approved therapies for these patients right now, right? And we can't authentically say, hey, we're going to solve this problem, but let us try to figure that out together and answer these questions. And the only way we're going to do that is through development, right? Understanding the science, understanding sort of the disease better, and ultimately, hopefully, having therapy options as opposed to people going straight into a kidney transplant, because that's horrible. As you know, like in some places, it's like a 4-year waiting list for a kidney transplant. So that is not a tenable, sustainable way to treat people with the disease, right?
Help me maybe speak to our community for a second here. If we have folks who are signing up on Power and saying, hey, I'm interested in kidney disease, but I've never been tested for this. Like, what's the best way to think about starting to engage with the research as a channel patient?
Partly because you have groups like NephCure and others. I think, one is, of course, we have patient advocacy, teams. We have people that they're welcome to interact with us or other sponsors. The other thing is clearly they should be engaged with their nephrologist to, what does this make sense for me? Should I think about this? And, proper education there. And then, of course, if they, sort of seem to be, individuals that should be genotyped, then of course, those are available for folks. And, I think the possibilities of trying to figure out, at least right now, they could be hopefully, in the category where they can be monitored over time. But if they have more progressive disease, they may be eligible for a clinical study, of course.
What are maybe the near-term milestones that you're really excited about? And then maybe a little bit further afield that you're looking forward to in the work that you're doing?
We have some important critical data on our APOL1 program that's coming later this year, which, if we hopefully see the data we hope to see, Brandon, the idea is then we could be in a registrational study, which is one of the last steps, of course, to actually getting an approved medicine. That would be tremendous if we can actually transition to that. That is the goal of all of our companies, of course, as biotechs, is to actually hopefully have an approved medicine. And then we also have a couple other programs using that same approach of genetics, variant functionalization, drug discovery. We had some data last September, which was pretty well received by the community and investors that we had on a second program utilizing the same techniques. We'll be starting two phase 2s based off of that data this year. So hopefully if we can advance those particular programs as well, you're talking about, a few options or opportunities that allow us to do what we hope to do, which is try to eventually get to an approved medicine and, be in our mind, part of that next group of companies that can be truly that next wave of precision medicine companies in the industry, right? That's our goal. That's our aspiration. Of course, a long way to go there, but I think that's what, we intended to do from the beginning, and we're built for that.
Tell me a little bit about the other condition areas that or I guess specific indications that you're thinking might benefit from this kind of platform and this kind of approach?
Yeah. So I think what's interesting, so we're always pretty authentic and genuine. Like that approach doesn't work for all diseases, right? It sounds like, oh, that sounds great. Why don't you just do this for everything and neurosciences? But what we rely on, Brandon, is, that data set that has genetic with paired clinical in a longitudinal fashion with a biomarker that translates very well to patient outcomes that is, understandable. So what's nice about kidney is you have that. You have protein in your urine that can be tracked over time that translates very well to kidney filtration, or that eGFR, which is a true measurement and quantitatively of proper kidney health. That's great. We need to work on diseases that have that. So there are other diseases, of course. So metabolic syndrome, you could also have markers that do that. Some forms of cardiovascular disease where you can look at particular biomarkers that are highly indicative of outcome of patients, which is ultimately like cardiac output, type measurements. So when diseases have that sort of like nice fine line between what we call the clinical endophenotype, or just the clinical biomarker, to an outcome, that works very well. Where it doesn't are diseases where that marker is exploratory, doesn't really correlate well. So take, for example, people ask us all the time, pharma companies, why don't you do this for Alzheimer's? Why don't you do this for Parkinson's disease? Well, the problem is the biomarkers, including some that have emerged in the literature, NFL or filament, ligand kind of measurements, that's great, but it doesn't really correlate to outcome. Like we don't know if that really has an impact by measuring those markers, whether or not it's going to impact, sort of the disease state or even outcomes of the patient. And so our system and our process and our data sets, we would not be good at doing that because it's not correlative enough.
Is that purely just a lack of data?
Or it's also just a lack of understanding of disease, right? I think for Alzheimer's, just to kind of build on that, everyone has kind of gone with the A beta or tau approach of forming aggregations in the brain. Yet even in these larger studies, even when you disaggregate sort of those buildups and plaques, it doesn't seem to change that much in terms of trajectory of disease. And how do you measure that earlier in the course of as they're progressing, which is not a biomarker like NFL, right? So I think that's the issue is like you would have to have a better understanding of overall outcome, pathophysiology. And that's why we end up working on diseases where that connection is more better understood. And now you have the data sets. So it's not just about data. You actually have the data sets plus a good understanding of pathophysiology that allows us, oh, this is a real signal versus some of the neurological diseases. Unfortunately, that signal is maybe there, but it may be misleading.
Who does the work to get those signals?
I mentioned the Broad. A lot of our geneticists were trained by those people on those papers. Some of them wrote those papers. So, we have people actually wrote papers on this concept here. And then we have we've built a really strong, we could go from target discovery all the way to the clinic. So, we built, sort of a group genetics, biology, drug discovery team that is fully integrated. They're all here in South San Francisco. We don't have a virtual kind of setup. They're all here interacting. And that was important to me because it was, how do you integrate all these disciplines such that you can fit for purpose, do what we're trying to do in certain diseases, right? So we have a great R&D group that's been built out. And then to complement that, drug discovery team that have worked on, not just concepts that got to the clinic, but eventual approvals, because having proper drug hunters is important, that know what a medicine looks like and be able to kind of guide in principle, okay, this has a good shot, at least, getting to a data set and maybe even to an approved product one day,
Absolutely. As we kind of wrap, I've got maybe a few hot takes I'm going to throw your way and just kind of curious what you think. Where do you think this genetics-driven approach is being over-applied today? And where do you think it's being under-applied?
I think if you go back to what it might be a little bit early to apply it to places where we don't really understand the biology, the disease, and pathophysiology. Because one, if we don't understand that, what are we actually looking for in signal? And then two, to your point, do we even have enough data to actually look for these particular things and generate sort of the functional understanding of what the genes may be doing, right? Like, we worked on ALS for a little bit, and, and it's interesting because it's clearly high unmet need, but the problem is, there's, I guess, one approved product, but it doesn't work very well. And could we apply our principles? The problem is very small data sets, right? And then, of course, then you run into the issue of the biomarkers not necessarily translating well to outcome because we don't know enough. There's only, one thing that's approved. So I think it might be at this point a little bit early and a little bit overused in diseases where you don't have that relationship. I think where it's underused is these bigger topics, like which is happening now. So cardiovascular disease, that would be interesting. Obviously a lot of progress in ATTR-CM as well as HCM, these acronyms, I'll strip later if people care. But, diseases that we have a better understanding, but larger diseases of heart failure, we don't have a good understanding there, right? And I think it would be nice to, again, can you identify certain individuals that might benefit from this? And then I think the other part is that obviously a lot of the first wave in terms of how you think about metabolic syndrome, a lot of success, of course, with, sort of the work at Lilly and Novo and others. But then, of course, what's the next wave of that is most likely going to be, not everyone responds. Can we understand why? Oh, these individuals have bone density loss, or these people have muscle loss. Can we understand that? Like, what's driving that? Is there a biological basis, or is it biological and genetic? So I think, there's going to be building off of what is clearly benefiting a lot of people, but it will have implications. I know we already know that, some of the GLP-1s have an impact to muscle loss, bone loss. So even with all the benefits, there are, issues with that. And so I think the idea that could you apply this approach to that and better understand like why people are responding or who's not responding? Is there another mechanism that can complement that in a more targeted fashion? Could be interesting using that.
It strikes me that your approach is very rooted in historical real world data. At the same time, there's a lot of oxygen being sort of in the room. You mentioned AlphaFold, AI applications to modeling new candidates. Is that all hype, or what is the viable path forward there?
It might be a little bit. So will it have a role? Yes. Do we use some of those techniques? Absolutely. Like we have, we use natural language processing on doing some of our things like variant calling. We use things like AlphaFold and other things. We incorporate that into our process, right? Now it is, does it actually replace, like, does that, like, even on this, like, let's take the AlphaFold example. Does it replace all structural biology and X-ray crystallography and cryo-EM? No, absolutely not, right? There's certain proteins that don't allow itself to be using those techniques in order to better understand at the resolution to actually drive your drug discovery, right? And over time, maybe it'll get better, but there are limitations with it. So I think for us, what we found is like integrating both of those is great. Can you actually more like reduce the cycle time by using a computational tool with a classical discovery tool like cryo-EM rapidly so that you can better inform the chemist on how you should actually be interacting with the pocket that you're trying to drug. And that's working, right? But it's still early. There's people trying to do like the holy grail of let's predict tox. Let's predict like ADME PK, which is like
Just jump to the finish line, yeah.
Yeah, now that's just really early. Like there's companies doing it and I think they're making progress for sure. Is it prime time to work across all target classes? And no, of course not. Over time that might get better, and it may not, right? It ends up being not just like having the right data, but also just, is it truly going to be able to like do something better than just having classical people doing ADME PK, right? We'll see. So I would say like that's still a little bit in the hype area, albeit we use a lot of those principles.
What can you do today using these principles, like in your day-to-day operations and the way that your kind of teams run that were not possible before? How is it showing up?
Yeah. So I would say, because we like systematically went through our process and like ran basically experiments on how to use some of these things, right? Like high throughput screen, like, okay, there's target identification. There's target validation. There's high throughput screening. There's lead optimization. Like we went through everything and looking at various tools where we find it, so variant calling is very helpful. So like the issue with variant calling, whether it's pathogenic, most variants are benign or don't do anything. And then which ones are protective? And the problem is you can have thousands of variants for a gene. So to try to do that, all of that experimentally, that's laborious, tough, and, takes a long time. So, can we use, like I mentioned, natural language processing to kind of like accelerate the process of variant calling to identify which ones are pathogenic or protective. Like that's been cool. The other angle is structural biology. So not all targets are amenable to this, right? So, what we end up, kind of seeing is can Rosetta Fold, AlphaFold, or any of these other bolts or these other algorithms, are they good at helping predict, whether or not what the pockets look like that we're interested in drugging relative to something like cryo-EM or X-ray crystallography, or even a more basic technique like photoaffinity labeling, like stuff like that. But combining it is helpful, but not one in isolation, right? So that's been helpful to better because at the end of the day, you want to be able to understand how you're drugging the target. The other area, we've looked at the entire, at least for us in our workflow of drug discovery, things like machine learning informed DELs and other things like that. They're informative for like I would just say hit expansion for people to understand. You kind of do a high throughput screen, you try to expand those hits, and then you move ideally into what's called lead optimization that ultimately results in your drug candidate. And we found it the most pragmatic, at least in our workflows, in hit expansion. It wasn't really great at high throughput screening. It was definitely not good at lead optimization. But where it was good, it was like, okay, how do I think about the hits I'm getting, and what do I think about the libraries I can explore in hit expansion? That was like a good tool. Right now it's not that great. Like I know there's companies out there doing it like Inductive and others, looking at ADME, PK, looking at those kind of, I think Lilly started a whole new effort just to look at ADME, PK, which I think is great. Like they should do that. But right now those aren't great, right? And then tox prediction, I'll forget it. So I would just say like, that's what everyone should be doing, is systematically looking at their own workflows about how AI and machine learning can augment each of those particular steps in the process. And then realize like some of them are great, some of them are augmentative, some are complementary, and others are frankly not there yet. And that's okay.
We couldn't agree more. We're doing it every single day here at Power. And it's funny. It's kind of like Wi-Fi on planes, in that it didn't exist. It didn't exist last year. And how bored are we already that it can already do parts of these things that they can do?
Absolutely. I mean, too, when you expand, because we're using it not all parts of the business, not just R&D, right? Like it's how do you do, things that you can do in terms of, hey, I want to pull all these markers research. I want to better understand the disease. I want to understand the competitive landscape. I want to augment how we do reviews, 360 reviews. Like AI is embedded all over the place in the org. They're really being able to, and one day maybe we can use it for, and I know people are looking at it, of course, like how do we optimize things like processes, like regulatory filings, amendments, like that's huge. That's going to save a lot of cost, time, translation work, as you're thinking about global studies. Like there's some super pragmatic things that definitely we're using and helping. But you just have to look through your workflows and your processes and see where it's real and where it's not, And that's okay. What I like is doing a bunch of these experiments as you do. Like I will have a vendor or we'll run an experiment. If it's better than what we're doing today, sure.
Why not?
Why not?
That's a whole other podcast series that we should probably do, which is AI in biotech. I feel like all day long I get hit with AI in software, and whatever. Like it's always AI in software, but I'm really curious about AI in life science. This is an awesome topic to kind of conclude on.
It's real, but it's a little overhyped, right? That's in that bucket, and it'll get better. I don't have doubts about that.
We'll revisit it in 3 months, and I'm sure it'll be adequately hyped.
There will be another version of it.
Just another breakthrough. Right. Yeah. Well, Jason, I've so enjoyed this conversation. I know we've overstayed our welcome a little bit. Thank you so much for coming on the show.
Yeah, no, Brandon, thanks for having me. All the best, and appreciate your time. Thank you.
Of course.